Okay, now what is oogenesis? Genesis means formation. Oocytes, formation of oocytes. Okay, the process of formation of mature oocytes from oogonia. Okay, one thing you should know that the mature oocyte is ovum, that is haploid, whereas oogonia is diploid. Okay, from where these oogonia or the spermatogonia in man they come from the primordial germ cells. We had read about that. Let's revise. Oogonia, they arise from primordial germ cells which are formed from epiblast cells in the second week of fertilization. From the epiblast cells, the primordial germ cells, they migrate to the yolk sac. Okay, and from there, at the end of fifth week, they arrive at genital ridge or the developing gonads, that is testis in males and ovary in females. Okay, so first of all, oogonia. Oogonia is formed by the mitosis of primordial germ cells in ovary, as I have told. Through the mitotic process, it converts into primary oocyte. Okay, both oogonia as well as this primary oocyte, it is diploid. And you should know that these primary oocytes, they are not formed after birth. Okay, they are maximally they are reached at the fifth month of intrauterine life, and maximum number of germ cells are 7 million at the time of fifth month of intrauterine life. Then these primary oocytes, they start undergoing meiotic 1 division, but they are arrested in the diploteny stage of prophase of meiosis 1 and remain in this stage till puberty. Okay, but primary oocytes will not be formed after birth. Okay, so one more thing, this primary oocyte, they are covered by a layer of flat epithelial cells. Okay, these are squamous cells. These are called as follicular cells. Okay, single layer of flat epithelial cells is there till puberty, and this whole structure, this is the primary oocyte inside. This is primary oocyte. This whole structure is called as the primordial follicle. Okay, so as I have already told that maximum number of germ cells, they are reached by fifth month of intrauterine life, and they are 7 million in number. And after that, most of the germ cells, they start atresia, they start degeneration, so that at birth only 6 to 8 lakh primary oocytes are there, and atresia continues in childhood till puberty. Only 40,000 germ cells remain. So I am repeating again that all the primary oocytes, they are surrounded by single layer of flat epithelial squamous cells, and this whole structure is called as primordial follicle. Here you can see this is the primary oocyte, this is its nucleus, this is the layer of follicular cells, that is single layer of flat epithelial cells, and this is the surrounding ovarian connective tissue. Okay, now but what happens near the time of birth, all the primary oocytes, they have started prophase, but instead of proceeding into metaphase, they enter into diploteny stage and arrested there. Okay, so they do not finish meiosis 1 before puberty because of oocyte maturation inhibitor. This peptide is secreted by these follicular cells, and it inhibits the further division of this primary oocyte from prophase to metaphase. Okay, hence you can see here, this we had already discussed. Okay, but this time we are just taking it as a revision. So this is the primordial follicle surrounded by flat epithelial cells. What happens is at puberty, some primordial follicles, they start, about 15 to 20 primordial follicles, they start maturing, they start growing, and this is the early primary follicle or the growing follicle. Here these flat epithelial cells, they convert into the cuboidal cells, and now they are called as granulosa cells. Okay, and during this stage an acellular glycoprotein layer start forming beneath this follicular cell layer or the granulosa cell layer. This layer is called as zona pellucida. This formation starts at early follicular stage, early primary follicular stage. On further maturation, this cuboidal cells layer, that is granulosa cell layer, it becomes multi-layered, okay, and the zona pellucida, it is well formed at this stage. Okay. So we should know there are two hormones we had read before also, follicle stimulating hormone and luteinizing hormone playing important role in this. Follicle stimulating hormone, it is not essential for the conversion of primordial to primary follicle, but in absence of FSH, these primary follicles, they become atretic and they do not go further. So this follicle stimulating hormone, it is required for in their maintenance and their further maturation. Okay. So from the mature primary follicle stage where the zona pellucida is well formed, what happens is in the next stage follicular fluid starts accumulating in the space, and the space is called as antrum. Also the this ovarian connective tissue around this, it starts arranging around this follicle and is called as theca folliculi layer. Okay. And further this stage is called as the antrum has started, antrum is forming, so we will say it is vesicular or the antral stage. Okay. This vesicular stage, it further converts into mature Graafian follicle under the influence of luteinizing hormone. Okay. Luteinizing hormone has mainly two functions. First, it converts the primary oocyte to secondary oocyte, that is, it helps in the conversion of primary oocyte to secondary oocyte. Secondly, it helps in conversion of this vesicular follicle to the mature Graafian follicle. In this mature Graafian follicle, the antrum has become well formed, and this secondary oocyte, it has got off center totally and is surrounded by a mound of these granulosa cells here. Here this mound is called as cumulus oophorus. Okay. And also the theca layer around the vesicular follicle, it further arranges into two layers, theca interna and theca externa. Theca externa is fibrous, whereas theca interna is secretory and is vascular. Okay. So actually what happens is just 36 hours before, 24 to 36 hours before, there occurs a rise in the this luteinizing hormone, which is called as LH surge. Okay. And what it does, this luteinizing hormone, it converts the it completes the meiosis 1, it helps in conversion of meiosis 1. So primary oocyte is converted into secondary oocyte and the first polar body, and the first polar body is extruded 24 hours prior to ovulation. Okay. After this secondary oocyte, it starts cell division further, but again 3 hours before ovulation it arrests in the metaphase and completes meiosis 2 only after fertilization. Okay. That is, meiosis 2 will complete only if the this secondary oocyte will be fertilized. Okay. Otherwise it will degenerate after 24 hours of ovulation.